Hey guys, welcome back. This is Lei. In the last video, I showed you how to set up environment for RNA-seq data analysis. In this video, I'm going to show you how to quantify gene expression from FASTQ files. The actual workflow only contains 3 steps. Step 1, adaptive trimming. Step 2, reference genome alignment. Step 3, gene expression quantification. But before that, we do have to prepare our data, which includes FASTQ files, genome index, and gene annotations, as well as some data QC at the end. There's not much to say if you already have FASTQ files on hand. Just put them into a properly named folder, and that's it. But if you don't have any, we can download an example dataset from NCBI GEO database. If you've never used GEO, just go search the GSE series number, and at the bottom of the dataset page, you can see we can directly download gene expression matrix from it. But in this case, we need the raw FASTQ files. Just go ahead, click on SRA Run Selector. On the next page, you can see the samples we're going to download here. There are 4 samples in this dataset, and they're divided into 2 groups. We can download them with our command line tools. So just open your terminal, activate the RNA-seq environment we created last time. You should have already installed everything you need for the tutorial. But here I'm introducing you a new environment manager called Mamba. Mamba is newer and faster than Conda, and it's already included in our environment. You can go try it. And now we go to the download folder. The tilde here represents the home directory. Most of the time, when you open a terminal, you're already inside of the home directory. Now let's go to the download folder and download our FASTQ files using FASTQ-dump command. We have 4 samples. Now we download them 4 times here. Once it's downloaded, we compress all the FASTQ files using gzip. And here, this command means we want to compress all the files in the folder that end with FASTQ. So the resulted folder should contain the following files. You can see each sample has 2 FASTQ files. That means this is a pair-end experiment, R1 and R2. That means the RNAs were sequenced from both 5' end and 3' end. So inside of a FASTQ file, there are entries. Each entry has 4 lines. The first line is the ID, second line is the actual sequence, third line, ID again, fourth line is the QC score. It doesn't matter if you understand it or not, because it's not meant for human to read. Next, we prepare the genome index and the gene annotations. First, we download gene annotation file for mouse because we have a mouse dataset. We go to GenCode and download the GTF file, which contains all the detailed information about the gene in the genome. Next, we download the pre-built star index for mouse. Well, star is the aligner we use here to map the reads against our reference genome. So you go to RefGenie, find mouse star index, and download everything in the red box and put them in the folder. You can, of course, build your own star index, but why? So when learning programming, I want you to be as lazy as possible, do as little as you can. If you can find anything online, just copy and use it. There's no plagiarism, right? But if you do want to customize your genome index, this is how you do it. So this is the standard format of how to use command line tools. First, we call the command, and then we set the parameters using the options here. We separate the command and options input and output files by spaces here. So we call star first. We want to use 12 threads. We want the RAM size of 200 gigabytes. We want to make a new genome index. We want our output directory to be this star grcm38. Our input is a FAST file, and we need a GTF file, which is the one we just downloaded. So with all the files on hand, we can put them into their corresponding folders. We can create these folders using command mixture. Remember mixture from last video? So here we use it. Mixture -p, the folder we want to create. -p means we create the whole path. For example, we want to create a tutorials folder in the home directory, and RNA-seq inside of tutorials folder, and then a star index mm10 folder inside of RNA-seq folder. So with one command, we can create a series of folders, right? So we create folder for our genome index, create one for GTF, create one for the FASTQ files, and one for trimmed results, and one for aligned results. And then we can move all the files using the move command. The format is move folder files in the old folder to the new folder. So with all the data prepared, we can finally start processing our data. The first step is to trim the adapters. We use trim Galore. Okay, the same logic we call trim Galore. Set the parameter FastQC. That means we want to do QC at the same time. Paired, our data set is paired, pair and data. We want to use 8 cores, 8 CPUs over here. The input has 2 FASTQ files, R1 and R2. The output should be in this folder. We just made this folder, right? So the resulted folder should have the following files. So among them, the FASTQ.gz files are the trimmed FASTQ files we want to use for the next step. So in the next step, we want to align the trimmed FASTQ files to our genome. We use star. Again, we call star. We specify the genome directory we just made over here. We want to run using 20 threads. So the input files are the trimmed FASTQ file 1 and file 2. We want the output file to be in the BAM format and to be sorted at the same time. We want the unmapped reads included in the BAM file, and we want the output to be standard. And our input is in the zcat, the compressed format. And lastly, name your output file, BAM file. So this is how it looks in the folder. So this BAM file is the one we want for quantification. So the last step, here. So this is how it looks inside of BAM file. It's quite complicated. Again, it's not meant for human to read, so just ignore it. So next step, we quantify the gene expression using the BAM files from last step. Here we use another tool called featureCounts. We want to use 8 cores. We want a gene level quantification, and we want the gene symbols in our expression matrix instead of gene IDs, because it's hard to tell which is which. And our data are not stranded, so we set 0 here. -A is the gene annotation file. We specify the path of the GTF file. We want the output to be a gene.txt file. Specify the output. And our input is a BAM file from the last step. So this is the result of our expression matrix. So you can see the first column is gene ID, gene symbol. Second column, chromosome. Third, fourth are the coordinates of the gene in the genome. And this strand, this is the length of the gene. Last is the counts, the gene expression. Okay, So these three steps, with 3 steps, we quantified one of our samples. We have 4 samples, remember? So just go ahead, repeat the 3 steps for all your rest samples, and we will have like 4 gene matrix like this. But they will be all in the same format, same order, but there are only 2 useful columns, the first one and the last one. The last one is gene expression value. So you can combine all 4 matrices into one consolidated gene expression matrix. It's very simple. You can do it manually with Microsoft Excel. So last, we check the quality of our data and the mapping rates by calling MultiQC against the whole folder here. And it will generate an HTML file that you can open in your web browser, and you can visualize the result. In this column, you can see the alignment rate is quite good. It's like 82%. And the assignment is also good, 60%. So usually we're expecting 80% of alignment rate and like 40%, over 40% of assignment rates, and both of them are good. So that's about it. And we have our gene expression matrix. And now you can go to my website, go to the second tutorial, copy my code, and try it on your own. In the next video, I'm going to show you how to perform D analysis with our gene expression matrix.